Good morning, everyone. I am Paola Ganugi from the University of Turin. And first of all, I would like to thank all the organizers for this opportunity, to present our work, which is entitled Phosphorus-Driven Morphological, Biochemical, and Metabolic Root Responses to Drought in Quercus species. So, when we think about drought stress in plants, we often focus on the leaves because they are visible and they quickly reveal stress symptoms. However, drought responses in plants can be easily tracked through changes in osmotic potential, stomatal conductance, transpiration rate, and chlorophyll fluorescence. Drought stress is first perceived in the soil, where the roots sense water shortage and nutrient availability. So we have to say that both organs are important to study plant resilience. Specifically, leaves reveal how the plants regulate carbon assimilation, adjust protective pigments, and reflect the nutrient status of the plant. While roots, which are in the soil, are the first drought sensor. They are responsible for both water and nutrient uptake, and they adapt form and function to soil. So only by combining this all kind of information, we can obtain a global picture of drought stress response in plants. Among soil nutrients, phosphorus has a crucial role in increasing plant tolerance to drought stress. Moreover, the phosphorus availability in the soil has significant implications for the competitive interactions between invasive and native plant species. Generally, low fertility forest ecosystems with low level of phosphorus in the soil are resistant to plant invasion, although some plants can acclimate by relocating phosphorus in order to supply phosphorus metabolic needs, thereby maintaining photosynthesis. While high fertility forest ecosystems with high levels of phosphorus can reduce the competitive advantage of invasive species, limiting the spread. The Northern American Quercus rubra, which is the red hawk, has colonized many forest ecosystems by replacing the original vegetation. This happened also in Italy, in the north of Italy, in the Po Valley, which is a zone which was once dominated by Quercus robur, the English rock, and now it's colonized by Quercus rubra. Quercus robur and Quercus rubra represent a very interesting model because they are very different. The native plant, which is the European native species Quercus robur, is very sensitive to drought and nutrient scarcity, and it has a lower resource use efficiency. While Quercus rubra, the alien species, has a lower nutrient and water demand, and it has an higher resource use efficiency. So their contrasting strategies make them ideal to study how drought stress can interact with nutrient availability in the soil. Basing on that, our question was, how does soil phosphorus shape drought responses in native versus alien hosts? To address this question, we conducted a greenhouse experiment, and the soil was collected from La Mandria, which is a natural park in the north of Italy, close to Turin. And this is a very particular soil because it has a very low level of phosphorus. We tested two water management, which were the well-watered condition and the drought stress condition. And we tested two different nutrient availability, the original low level of phosphorus and the phosphorus enriched level. Our measurements included some analysis on the leaves, like stem water potential, chemical characterization, and biomass, in order to give a complementary picture to the above ground parameters. While on the roots, we calculated biomass, we performed the chemical characterization, and we did metabolomics and FTIR spectroscopy. So results, stem water potential. Both species under well-watered conditions maintained high values of stem water potential, which was around minus 0.5 megapascal. which is in the blue circle, while both species at the end of the stress period reached a value which was around -3 megapascal. Biomass allocation. So both species allocated more biomass to the roots when compared to the leaves. This means that this is a structural trait of their growth strategy and not a drought stress-induced adjustment. Especially under well-watered conditions, high values were observed for the English oak, so for the native species, compared to the alien species. So we can say that the English oak developed a larger root system than the red oak, and especially under drought conditions with a phosphorus supply, the English oak root biomass increases, while for the leaves, higher values were recorded for the red oak. So we can say that the red oak balance between organs shift toward the leaves. So these contrasting allocation patterns reflect different adaptive strategies. In particular, the English oak showed a great root development, while the red oak showed a great biomass allocation to leaves. This plasticity in biomass partitioning is a well-known plant mechanism to optimize the resource capture. And so the great development of root by the English oak and the great response of this species to phosphorus supply suggests a way to cope with environmental constraints. So regarding the leaf and the root chemical characterization, I would like to present results about the phosphorus because they are the most interesting findings for us. So both species allocated more phosphorus to the leaves than to the roots, with drought reducing root phosphorus content. But under low phosphorus supply, the English oak accumulated more phosphorus in the leaves than the red oak. While with phosphorus supply, the English oak stored more phosphorus than the red oak in both organs, reaching higher values under both well-watered and drought conditions. The FTIR spectra told us how the interaction between drought stress and nutrient availability can affect root chemistry. In particular, we observed that drought reduced the content of saccharides and lignin-like compounds in the English oak. These are energy-rich and structural compounds, while drought in the red oak increased saccharides. But under drought conditions with a phosphorus supply, the English oak showed more stability, while the red oak increased saccharides, but the aromatic compounds were less. So this suggests a weaker defense for the red oak. Metabolomics analysis was conducted with an ultra-high performance liquid chromatography coupled to a mass spectrometer. And with this analysis, we identified more than 900 compounds, which were both primary and secondary metabolites. These results confirm the previous results, because we saw that for the primary metabolites, drought repressed both amino acids and carboxyl acids, but the phosphorus mitigates this effect. The phosphorus is the yellow bar, while in the red oak, drought stress repressed amino acids and carboxyl acids, but the phosphorus supply amplified this effect. Regarding the secondary metabolites, the water deficit downregulated the accumulation of secondary metabolites in both species, but in the English oak, the phosphorus supply mitigated this suppression, while in the red oak, the phosphorus supply leads to the downregulation of both isoprenoids and phenylpropanoids, which are involved in the plant resistance. So this work has a very important ecological meaning. The red oak has a conservative strategy, it's not sensitive to drought stress, and that's the reason why it's invasive. While the English oak, which has an higher resource requirement, and it's very sensitive to drought stress, and that's the reason why it's easily replaced by the red oak. But with the phosphorus supplies, the red oak loses stability, while the English oak gains resilience under drought. So, to conclude, roots are the primary site where drought and nutriodynamics interact. The phosphorus can affect biomass allocation, nutrient partitioning, and metabolic pathways. Quercus robur and Quercus rubra have different strategies because Quercus robur shows plasticity and benefits from phosphorus applied, especially under drought, while the Quercus rubra is stable in the poor soil, but it's destabilized by the excess of phosphorus. So fertility can affect plant function, but also interspecific competition, and so forest resilience. Thank you for your attention.